Hey, everybody, and welcome to MBF's latest webinar. Do you really need a light sheet microscope for brain-wide analysis, where we'll explore using serial sections for brain-wide analysis. And I'm thrilled today to have with us an expert in neuroanatomy and longtime MBF collaborator, Dr. Chip Gerfen. Hi, Chip. And also our Director of Machine Learning, Dr. Brian Eastwood. And I'm Nate O'Connor. I'll be emceeing things, and toward that, just a couple of small logistical things for the webinar today. Feel free to ask us questions at any time. All you need to do in the interface of Zoom is click the Q&A button, and it'll open your question and answer panel. Then you can just type your question and press enter. So I want to thank you for joining the webinar. And just a little note, if you have any questions that we don't get to, and we'll try to, at the end after we talk, we'll have a dedicated Q&A session. But if we don't get to any, we'll have your emails, and we'll try to get to what we can. But you can also always go to our forums at forums.mbfbioscience.com. And also, you'll get a short survey after this webinar. Please take the time to fill that out and let us know how we did. With that, I'm going to stop screen sharing and pass it over to Chip and Brian.
Thank you, Nate, for the nice welcome. And I'm happy to join you for this webinar to describe NeuroInfo, which is my favorite program to work with that we've been developing for some time now. For over 40 years, I've been mapping connections in the brain, and the one thing that hasn't changed is the necessity of analyzing circuits through the whole brain. One thing, and many things have changed in that 40 years. A major thing is that the neuroanatomical methods, we thought that they were fantastic 40 years ago and kept improving, but in the last 10 to 15 years, the advent of molecular genetic techniques and viral techniques have just exploded in terms of our abilities to map very precisely specific connections. It's a very exciting time. Another thing that's been a major advance in the last decade or so, or a little longer than a decade, is the ability to map these connections into the whole brain and the standard brain atlas. This was an approach that was developed. We have to give big credit to the Allen Institute, because they were ones that really pioneered this when they mapped all of the genes in the brain and they put it into a standard format and into a standard atlas. And then they did the mouse connectivity project, which again, in fact, we've got some poster related to that in the background. What that enabled was being able to map connections, or the projections of from 1,000 different injections in 1,000 different animals, and combine them into a single atlas so that you could compare between cases. This was an incredible advance. Of course, the technology behind that and the difficulty of doing that was something that institutions like the Allen Institute and others could do because of the manpower and the brilliant people that they had doing these projects. What NeuroInfo does is it actually brings that technology into individual labs, and so it provides a program that actually can mimic and do quite closely the same type of mapping to a standard atlas that is done in the Allen Institute and other places, other labs like that. So what I'm going to describe today is using the NeuroInfo program to map serial sections into the standard atlas. Now there are many there are many histologic techniques that may be used for this sort of thing, and one of those is using the light sheet imaging, so that the whole brains can be imaged. And actually, I was going to say that one of the things that has occurred over the last during the time that I've been doing neuroanatomy is that the processes have become much easier in terms of being able to map whole brains and to do clearing and light sheet microscopy has really accelerated the ability to do whole brains. However, being an old-time neuroanatomist, there's major advantages to using immunohistochemical processing of individual sections, free-floating sections, because we can apply, 3 to 4 to 5 different immunohistochemical probes to those and look at those in individual sections because they've been free floating. So what I wanted to do today is describe how we use free floating serial sections that have been processed for immunohistochemistry to be able to register those into the Allen Atlas. What we're going to show in the webinar today is how to use NeuroInfo to produce the first step of being able to register serial sections into the Allen Brain Atlas or into a standard atlas. The first step of that involves taking serial sections that have been processed with immunohistochemistry and putting them into a 3D volume. I'm showing here a single section through the brain from one of those, and I can scroll through, go to different, and scroll through that brain from the front. There's an injection site I'm going to describe later, all the way through. So this is the case that I'm going to show you how we reconstruct this. From the front of the brain, all the way through. So this is an experiment in which what we've done is we've used the modified rabies technique. This is a technique that was developed by Ed Calloway and Ian Wickersham that's been spectacular for tracing connections in the brain. What this involves is that there's a modified rabies type that can bind to a bird virus receptor, rabies bird receptor. And the way that they've developed this technique is that to have the bird rabies infect neurons, you have to actually express the bird receptor on specific neurons. This is ideal because the expression of that bird receptor can be controlled and only expressed by neurons that express something like Cre recombinase. So viral vectors are made so that the bird virus receptor is only expressed on neurons that express Cre recombinase. In this study, and in other studies, we use brains that Cre recombinase is expressed, in this case, in D1 receptor neurons in the striatum. Those D1 neurons are in the striatum then the bird virus receptor only is expressed by D1 neurons. And then rabies, when it's injected, will only infect those neurons that have the bird virus receptor. And then they're transsynaptically transported from the neurons that are innervated in the striatum back to the neurons in the cortex that provide the projections to those. So this is showing the mapping where we've labeled with, these are all rabies labeled neurons in the cortex that project to D1 neurons in the striatum. But we've done a second thing, which is that there's multiple bird virus receptors, and this is a second one that binds to a different bird virus receptor. And in this case, what we've been able to do is infect the D2 neurons in the striatum with the second type of bird virus receptor, and then inject the second type of rabies virus. And so we can label, in this case, the D2 neurons, and the cortical neurons project to those. And we can inject at the same, or label at the same time, the inputs to two different populations of cortical neurons that project to either the D1 neurons or the D2 neurons in the striatum. So that's the experiment that we've done. And what we'd like to do, and what we can do with NeuroInfo, is actually map the projections from the cortex and throughout the brain by processing these sections, serial sections. So I can just age up, go to the front of the brain, and you can see that the neurons that project to the neurons in the striatum are in the cortical areas all the way through. This is the injection site. There's neurons in the thalamus that project to the striatum and the amygdala. And this is why the necessity of being able to do whole brain imaging, neurons in the substantia nigra, pars compacta. So this is why, to analyze this type of circuitry, why it's necessary to do whole brain analysis. And we can do that using NeuroInfo. So this is the first step of that. I'm going to describe how we make this 3D volume for the next step, which is registration. Okay, so after we've generated that, the next step is that we use a cell detection method so that we can actually view the so we can mark the cells in the cortex. These are the cells marking the neurons that are labeled at the injection site and in the cortex. We zoom into the cortical area to show you how that's done. So here we can see that the get rid of the each of the neurons, each of the red neurons is labeled separately from the neurons that are green. And so in registering this brain to the Allen space, what we're really interested in doing is registering the location of all of these neurons that we've detected throughout the brain into the Allen space. So the next step of this, and we're not going to show that today, but it's the seminar following this, which is what we're going to show is how these neurons that are labeled in this brain are registered into the Allen space. Okay. After the brain the 3D volume has been generated, then we take that volume into the NeuroInfo, into the next step of registration, and we register those serial sections, and then we register them as a volume into the Allen atlas. So these are showing the location of the neurons. Those have projected the D1 neurons in the cortex, the injection site where the neurons at the rabies are injected into the striatum, and those neurons are labeled, and then the neurons are labeled. And the Allen atlas then is parsed into about 2,500 different structures that are all laid out. And then each of these structures, then the count of the numbers of each type of neuron are counted and actually provided in a spreadsheet that it contains specifically the number of neurons and other information about those labeled neurons throughout these 2,500 sections. We can go through and see that, the labeling, most of the neurons that project to the striatum are in layer 5. We can see how accurately the registration is. Highly accurate. Flip back and forth. And so this maps all of the neurons throughout the whole brain in the Allen space all the way through. So this is what we're going to be showing at the end of or what we'll be showing in the next webinar is how to get this registration. But the first step of doing that registration is we have to generate the 3D volume from the serial sections. Okay. Okay, so this is the NeuroInfo program, and what we're going to demonstrate today is the use of the serial section assembler. This is what assembles all of the sections that I've shown before into serial sections into a volume. First step of this, we click on that, and it generates, or it shows the workflow. So there's 6 different steps to this process. The first is we select the slide image files, then we edit them, finalize their section order, and go all the way through. So what I'm going to show today is, or what I'm going to show now is, how we do this work, follow this workflow. So the first step is we add slides. We add slides, and what I'm going to do is that I have to navigate to the location of those slides. So the slides that contain the sections from that case are in a single folder, and I select, there's 5 slides of sections that encompass all of the sections from that brain. So now it's loading them in, and it shows the 5 slides. So this is the first slide. And what we've done for this brain, what we do for these brains is we cut every section from the rostral pole of the brain through the cerebellum. And then we mount those, we process those sections for immunohistochemistry to visualize, various markers. In this case, the rabies, the red labeled rabies neurons and the green labeled rabies neurons. And we could mark other things like tyrosine hydroxylase, other immunohistochemical markers if we wanted to. And those sections, and after they're processed, are mounted onto slides, and and then imaged. And this is showing the first slide that's imaged, and you can see that's every section from the front and arranged in columns all the way through to the end of that slide. This is the second slide showing similar sort of layout. That's the first step, is just loading in the slides and seeing that they're arranged and are, actually this program allows that even if they're not arranged in perfect order, they can be sorted out in the next steps. The next step is that it's going to outline the sections, and Brian's going to describe that.
Yeah, so the goal here in this step is to identify every single section that's on each of the slides and set up the relative order on the respective slides. The software uses a process for edge detection to find the contours that surround each of these sections. And so the idea here is to identify contiguous regions that have strong edges around them, and that is what the software identifies as being a separate section. Sometimes some sections are a little bit close together on the slide, and in that case, you might want to split it, and as Chip is doing right here. And in that case, what the software does is it finds the best pair of points to split that section up into two contiguous regions. And also, in other cases, there might be a little bit of contrast on the edge of a section. I think there were a couple that Chip was working on previously. And sometimes there, again, you can just split that region off and delete it, or you can delete a couple of the points that are on the contour. Let's see, here's a good example. Yeah, so if you split that region, you can then delete the artifact.
Sometimes they're just pieces around, and then anyway, very quickly we can go through and do that. And then there are 15 sections on this one. On that slide, we go on to the next.
The other thing you can do is you can also adjust the parameters that are being used for the cell detection. I don't know if you want to pop into that, Chip, or not, but
yeah, that's a good point. So I'll do that in a second.
Okay.
Just cleaning up a few things. Looks good. So we'll go into the next section, and I'll. So one time, so as Brian was describing, the contours that are generated around these sections are sometimes, in this case, it does a very good job, but sometimes it doesn't do such a good job. And let me just. And so we can adjust.
Yeah, I'll talk about a little bit about what those parameters are doing. So they're controlling different components of this edge detection algorithm. The first parameter there is a smoothness. It looks at the scale of the edge in microns, so the difference between the background and the foreground. How large of an edge do you have?
And in this case, if we over adjust that, you can see that it's not distinguishing between the various sections. Move it back. Does a better job, but there's still some. Ideally, what we'd like to do is have it be very close fit around the edges of the sections. So these are the parameters that can be varied.
Yeah. The second parameter there is the threshold that just looks at the magnitude of the edges. And so adjusting that that slider can end up including or excluding different parts of the image.
So of course, to demonstrate this is a case where the sections are very easily detected by these detection, by this detector. But if you have some that sometimes you have problems with, this is the way that you can go in and manipulate those. So
yeah, the important part here, though, is that we just want to make sure every section's identified and surrounded by a contour before moving on to the next step.
Done editing that one, go on to the final slide. So sometimes there's sections that are partial sections, and so I just eliminate those.
Yeah, here's an example where you might need to use your detection settings. This looks like you're missing two sections in the
This is a very
slide.
good
So there you go.
So there we have to alter the detection settings. These two are missing at that point. Now it detected them. All right, so now we went through all of these. It says gone through editing and ordering the sections. And so we finished reviewing the unfinished slides. You want to go to the next step? Yes. What this next step is, and we're quite proud of this, how this works, is that this is actually a contact sheet. Most people are probably too young to remember. In photography, we used to use contact sheets, which was enabled looking at multiple negatives at the same time and arraying them. So what this does is it arrays all of the sections from those 6 slides onto a single display from the front to the back in rows all the way through. And so this is the first section from the first slide, second section from the first slide, first section from the second slide, all the way through. They're all numbered, so you can see how they're arrayed. And we can go through and you can check to see, okay, everything's there in proper order. If they weren't in proper order, this enables being able to move them around. So let's just take, a couple of these. I'm going to select a couple of these, and I'm going to move them. So let's say, so these two sections are obviously out of order, and I can pick them, cut them, move them back to their original position. You can do that for a whole range of sections. Sometimes you might get the whole slide that would be put in the wrong position. And move them all at once. Because they're numbered, makes it easy to move them around. So anyway, this contact sheet enables being able to go through and actually check to see how well, if they're put in the proper order. Like to say, we've been doing this, putting sections in proper order when we do the histology for some time, and so rarely make mistakes. However, it was when we first started this process, we were always making mistakes in terms of how they were mounted on slides in order. And so anyway, you can zoom in and say, okay, these two I know are fine. No, but anyway, this contact sheet enables doing editing at this point in terms of the sequence. So the next step.
Chip, before we go on to that step, actually, I wanted to take one look at this, at the contact sheet. And I wanted to point out that there are a couple of sections that are flipped one to the other. So for example, if you look in the third row, section 121 and 2-1, or those two sections, for example, the injection site is flipped from one side of the brain to the other side of the brain on the screen.
You can see that these are the two that Brian's talking about. So you can see that these, obviously these sections, when we mounted them, we mounted one reverse to the other.
Yeah.
So and this happens. So at this stage, it's not possible to flip them. However, that is done in this next stage, but that's something to look out for.
Yeah, I just wanted to make sure that we pointed it out before we went on to the alignment step.
Yeah, that's a good point. Yeah. Then we're going to go to the next step. So here we're going to do automatic alignment. And Brian, do you want to describe that?
Sure, yeah. Maybe what I'll suggest is that while I'm talking about it, you can just kick it off. So if you go ahead and do the align using the most accurate, then we can, then I'll talk about what's happening behind the scenes while that's running. So what we're doing here in this step is, the goal here is to start to reassemble the brain that was physically sectioned into a 3D volume. And the way we approach that is we use an image-based registration to align each section in the series to its neighbor. And there are 3 levels of registration that we can use for doing this, and they sort of trade off between speed and accuracy. The fastest option is to just align the centers of every section, right? And that is something that might be good to choose if all of your sections are in the same orientation as we just spoke about, or the same flipping. And that, if you're doing something like thaw mounting with a cryostat, that might be the case. In this case, I think you're using float mounting, and so that's why you end up with flip sections. And that'll basically, that first option will get you to an assembled volume really quickly. The second option is to align based on the shape of the section. So we use those contours that we found during the segmentation step to align one image to the next. And that option runs a little bit faster than the final option. The final option uses all of the information that's present in each section. It runs a full image-based registration. But this process has been optimized so much recently that I just recommend using that process all the time. And one of the things that is happening during this process is we're testing out the idea of flipping every single section relative to its neighbors to see if we get a better alignment. And if we do, then we say, okay, that section's been flipped. And so that's what that checkbox enables you to do. If you happen to know all of your sections are not flipped one to the next, you can disable that. But if that's checked, then we will be checking for flipping throughout this process. I think we're almost all the way through the brain, and so this is one of the parts where there's a lot of heavy lifting going on, and behind the scenes, a lot of computation that's happening. But as I said, it's been optimized a lot recently. We've gotten through this process a lot faster.
So the step before this, when we were going through and editing the sections, when the outlines were drawn, that requires some manual adjustments and things like that. And when we go through it, it might take 5 minutes or something like that. But then this step is completely automatic. So even though there's a lot going on in terms of user work, there's none. So now after that's done, it says the alignment is successful. Click next step to continue. This takes us to the next step. And this is the first section in in all of those sections having been put together into a volume. This is the first section. And so there are various controls. One is that I'm going to go on to the second section. I'm going to go to the second section. Nope, I'm going to go back to the first section. So this individual section, this is the first section there. We, as a matter of course, always put the side that's been injected on the left side. The brain, the rat's or the mouse's right side, but the image's left side. So I just did that flip. And then I can adjust by rotating. And I want to get it lined up And there's a grid function. You can put in a grid, you can change the size of the grid. I just use the grid to put some lines in there so that I can line up the midline. Once I've got that done, leave it on. I like to turn it off. And now we go to the next section. So this is now overlaying. This is the first section. This is the second section overlaying, and we can toggle back the transparency to go from one to the other. When I'm in the middle, I can now rotate. I'm using the mouse wheel to rotate the section. And now I'm holding down the mouse and I can move the section around. Line up the midpoint, do a little bit of rotation, get everything lined up. So for the most part, the automatic registration, the algorithm that the process that Brian has put into this program does a very good job, and there's little adjustment that needs to be made. However, you can just make minor adjustments just to see. I like to see that the vesicle or the blood vessels are lined up, one to the other, the midline's lined up. Usually does a very good job. What I do is typically just assure myself that it's well lined up, go through. And so now I'm going to just go section by section. And so this is another manual part of the process that's required. And after some time, you can typically, this is another thing that it only takes about 5 minutes usually to do that.
And I think here, the goal is to just make sure to check to make sure that the flipping, there haven't been any sections that have been misflipped. And you can adjust the registration if you want to. The other thing you can do here, next to the flip button, there's a refine button. You can run another round of registration for the pair of images that you're looking at. And that'll just start with whatever the registration is that you have right now, whatever that transform is, and it'll just try to run another optimization from there. Sometimes that can be really useful if you have like damaged tissue. You don't often have damaged tissue, but sometimes if you have damaged tissue, you can line up interactively line up the part of the brain that you want to be aligned, and then run refinement, and that'll snap those features into place.
And remembering from the contact sheet that the part of the brain, the sections that had the injection in them, some of them were flipped one to the other, but as we go through this, the automatic alignment has flipped them appropriately. So we don't have to reflip them. If it turned out that sometimes there was a case, if it had not flipped them, we would look at this, and we can actually change it here. Say, oh, the injection's on the left side here, and in the adjacent section, it's on the right. So all we do is we flip it, make more adjustments, and move on. So I'm not making any adjustments, and the automatic registration algorithm has done such a good job that you can see that there's actually very little that needs to be adjusted. So as we get close to the end, okay, only 2 more to do. Okay, now we're done. So now we go on to this one needs a little bit more. So now we go on to the next step.
So another great part of this program is the case file. And what this is when we've gotten through all that process.
In the background, we haven't described this yet, but the program is recording every change that we've made, everything, and it's recording that, and it allows us to generate then a case file.
That's right. So everything that you've done, from loading all of the slides in, finding all of the sections on each slide, setting up their order, aligning them, every single piece of that process has been captured inside this case file. And so if you want to come back to this experiment at a future time and change something, maybe you missed a slide, or maybe you find out that there's a section that's out of alignment or out of order, for example, you can come back in and make those corrections, not have to start completely from scratch, but get through to the final 3D reconstruction much faster.
Then the final step is that we can take those and save that reconstruction. And there's various options for how to save that. We can save it as a single stack of JP2, the individual sections, or JP2 images put in the stack, which makes a JPX stack. Also, we can save them as a series of images, where we could save the individual sections all aligned, and everything that's been processed through that is either individual JP2 or TIFF files. Some programs, and sometimes people want to bring this into other programs, Python, various programs like that, and this enables doing taking those sections that have been aligned, taking into other programs. There are various things that can be done. What we typically do is to generate a single stack and then hit save reconstruction. Not going to do that now because the images, or the case that I started off showing in this webinar, was what was produced by this process. What I want to do, though, is to close the program. Do I want to close the workflow? And the answer is yes. And then I'm going to close all of those images. Excuse me. And I'm now going to demonstrate this case file. That case file was stored with all of the original, in the same folder as the original slides were. I'm going to open that up. And what it's doing, what Brian just described before, was what it did is it loaded all of the first, it went to the first step, it loaded all the slides. It then did all the editing, and all of the minor changes that we made to these outline sections are all done. You can go to the next slide. It remembered all of that. This isn't trivial because oftentimes one might make a mistake and say, oh, I left out a section or whatever. You can actually add those back to this step. The next step shows the contact sheet. Applying all of the alignment transforms that were used before. Anyway, it goes through all of this. So this is a very useful part of the program, that if you make mistakes, or you come back, or you go through and say, this has happened to us. Sometimes we go through and say, somehow we missed a whole slide and we didn't put it in. And so we come back and we could actually add it using this case file process. Okay, so the final product is this JPX file. That's a stack of the serial images that have now been aligned one to the other all the way through the brain. And then, as I said before, the next step of that is what we went through, was to detect the cells, have them marked. You can actually visualize that in 3D. And then the next step, what we did was to take these and we're going to register those into the Allen space and make have all of the neurons that are detected in the different areas quantified. Okay, thank you for your patience and all. Happy to entertain questions.
Great. That was really, really fantastic, Brian and Chip. Thanks so much. We have plenty of time for questions now. So if you have any, again, just be sure to click on that little question Q&A bubble. And I've been trying to answer them as we're going along in the background here. And yeah, we have time for more. I think just to note, I had a couple questions that I answered about the next webinar. One thing we'll definitely email out when the links and the dates and everything for that first one listed here, the mapping brain images to a reference space. So we don't have a date or plan for that, but it will be soon, and that's where we'll really dig into taking what Chip showed today, making that brain volume and then matching that to a reference space. Okay, so we have a question, and Chip and Brian, I'll let you answer this. And the question is, what if some sections have splits or tears?
So Chip, you're actually muted.
Okay, so that's a very good question now that I'm not muted. And so yeah, so oftentimes, and it's the nature of doing histology is the sections sometimes get ripped or torn or things like that. And at this point, it's not possible to actually take the parts of those sections and put them back together in the NIMH program. So the best thing you can do is to be very careful when you do mounting your sections and all. It is possible that if there's it's possible that if there's a missing section that you can put in a placeholder for a missing section, so that if we go through and we drop a section, or you notice that there's a section missing all the time, it's missing when you do the processing. And it is possible through this process to add like a placeholder. And that's important because when we do the reconstruction and then register to the Allen brain as an example, it's necessary to make sure that that missing section is taken into account when we do the registration. And unfortunately, at this point, we're working on that. We've been working on that to find ways that we could actually take parts of sections and put them, virtually put them back together.
Yeah. The other thing I'll say on that is, that's when you have damaged tissue, sometimes it's really challenging for an automated method to do the right thing, but we have those interactive methods, right? That step where you review the alignment, you can make that decision and say, okay, this is the part of the tissue I want to align, or I can kind of figure out, what the average is that you can kind of place that section in the space to build a reasonably good reconstruction for it.
And that's exactly what
And the other thing I'd also mention is in terms of putting the sections in sequence and all, that's an important part of this. Actually, the program allows for mistakes to be made at that point, but on the side of processing, the better job that can be done putting the sections in the right order when they're mounted is the best thing to do. What we do is we use a 10-well plate that we put the sections into. And go, one section per well, and section go all the way down so that each well ends up with a, 1 in 10 series. And then we use that because it's trivial to put together, put in sequence sections that are in 1 in 10 series. But then we, when we do the actual mounting process, we actually put them all together in series. And again, the better histology and better, the better the histology, the better sections are going to go into this program.
That's great. So one question that came up while you were talking was specific about the case that you were looking at, Chip, and what it was cut at, and if it was intervals and what magnification.
Oh yeah, well, so our standard is that we take every section. So that's important because in the mouse brain it's not very big, and so there's actually, if you take one and even a 1 and 2 series, it's possible to miss important parts of the structure. So we routinely take every section. We cut every section. So the example that I showed was every section, and the sections are cut at 50 microns. And so for a brain, a typical mouse brain, it's between 100 and 120, 140 sections from the front of the brain through the cerebellum. Is about 120, 124, 40, 140 sections, 50 microns. And so we don't do, it would be possible to do every other section, but we do every section because it makes for much better registration to it's just better analysis all around.
Great. Just one quick question. This is kind of a logistics thing, but of interest probably to everyone. I've been asked a few times while you're talking, is there a trial of the software? There absolutely is. And a link to that will be in a follow-up email that we send you after the webinar to everyone who attends. Here, another kind of MBF specific question is, can you take a registered section to the Allen Brain Atlas and then use other MBF software probes such as Spaceballs or Optical Fractionator? And the answer is absolutely. What you can do is you could draw your regions, and we'll get into a little bit of this in the next webinar, but you could draw your regions, save them as an XML file, one of MBF's data file formats, and then simply import that in the image into Stereo Investigator and go from there. And we have a few folks doing things like that, doing stereological processing on things that have been automatically. So, and that really eliminates the need to have to draw, regions on tons and tons of sections. So I wanted to answer that one. This question came up a couple times too. I think it's a really good one, Chip. Are you using atlases other than the Allen CCF? What about the most caudal and rostral slices, which are not part of the CCF?
So for what I do is we use the Allen CCF because it covers most of the brain that we're most interested in. But there are other atlases that have been incorporated into this, and I'll let Brian speak to that.
Yeah, let's see. So for mouse, we have 2 different atlases. We use the Allen CCF, and we also have the one that we developed together that has a Nissl average brain. And then for rat, we have the Waxholm space, which is one of the standard atlases for rat. And I think, if I'm not mistaken, those are the 2 major atlases that are easy to get with the software. And then there's some others that we're working on getting online that I believe it's just a matter of licensing at this point. We have them technically working and stuff. So, but I think as far as like going further rostral, I don't think we have a model right now for anything that's much further rostral than like the olfactory bulb that's in the Allen Atlas. And then going further caudal, as you get into the spinal column, I don't believe the Allen Atlas goes all that far.
So basically, if it's not in the Atlas, we can't, there's not a lot we can do.
Yeah, we can reconstruct it. Like we
Right.
can build a 3D reconstruction, but we don't have a model, to do that last step to be able to put it into a common reference space.
And then make annotations in those sections.
Yeah,
that's right. The part that we showed today in terms of being able to reconstruct the serial sections, those can be done all the way through the brainstem. Whatever this program is very good for reconstructing, for registering. That's to be developed.
Yeah, and I believe other people have used BrainMaker for other organs. Even it's named BrainMaker, but people have assembled other types of tissue
for kidney, for lung, spinal cord, gut. Here's a question, Chip. I know you like this one. How long did this whole process take you from staining to final 3D volume construction? Just wondering about the time-taken comparison between this method and the clearing tissue plus light sheet imaging.
I like to say, it's myself and Ron Piletsky who works with me. We can process typically, we can process like 5 brains a week, 5 whole mouse brains a week. That's from cutting through, through imaging and everything. Excuse me. So, cutting a brain, frozen, fixed, frozen, takes about, half hour to cut a brain into serial sections. Then the histology for doing immunohistochemistry is, for the way we do it, we have a tray system where we can, transfer the sections from, through the different steps of immunohistochemistry. So there's nothing involved with that, but it takes, overnight for staining, but the manual labor on that is trivial. And then the real slow part is the imaging of the sections themselves. And that imaging, depending on, we image at 10x with 10x objective. And we found that takes, depending on, it takes, for us, individual slide, because we image through multiple planes through the section. That's important in terms of making sure everything gets, everything is properly focused and everything. So it takes, 2 to 3 hours per slide. And so we can image, or do the imaging on a full slide in, a little over a day and a half or whatever. And then this part of the process, that once we have imaged and bring it into the NeuroInfo program, what I showed today is basically the major manual part of it. It takes, as long as it took me to demonstrate today, it takes 15 minutes to do, the BrainMaker part in terms of manual, the manual part of it. And then it runs, the saving part does take about an hour on the computers we have, but so it's about an hour. So from start to finish for a single brain, it takes 2 to 3 days. And that's the amount of labor it's actually, the way we've got it worked out is pretty minimal. One thing I will say is that we can, for doing, because we do free floating immunohistochemical sectioning, we can, on the particular brain that I'm showing it, we can go up to 4 to 5 fluorophores, different markers in individual sections. So anyway, that's about the amount of time it takes.
And so we had another question. Just to clarify, the software reconstruction can be run not just on brain tissue, but it can also reconstruct things like organoids. That is correct. You can put in a series of images and reconstruct a volume of any organ. It's when you go to do the mapping to an atlas space, you need that atlas to exist. And right now we support rat and mouse. So just a little clarification on that, because that came through as a question again. Someone asked, do we have a workflow for mouse spinal cords? And again, you could reconstruct spinal cords. We have folks doing that, but getting to the registration to an atlas and the annotations and data mapping and such, we don't actually have an atlas for that. If anyone, if you ever know of one that's a digital atlas, there are some aspects to an atlas that are necessary to be able to integrate into a 3D registration system, but we're always happy to take a look at an existing atlas. Okay, here's a really good question, and it came up earlier, and I answered it in part offline. But they're asking about the registration of single coronal histological sections. Are we estimating all the angles that can happen around the dorsal ventral axis or the left-right axis? So Brian, I'll throw that one to you.
Sure. Yeah. So for what we covered today, we're just looking at reconstruction of a brain, so each section to its neighbors. But NeuroInfo has a couple of different registration steps that happen. And so if the end goal is registration to the Allen atlas or any atlas, then it's that section to atlas registration that you might consider the most important one, right? Because that's going to give you your anatomic constraints. And there, yeah, NeuroInfo's registration for doing either individual sections to the atlas or multiple sections to the atlas considers the sectioning angle. We have a full model of what happens when you are sectioning a brain physically on a microtome, cryostat, whatever you use. That is something that is incorporated into the NeuroInfo software. And when we do that stage of registration, when we're targeting an atlas, we model exactly what that sectioning angle is.
So that'll be covered very well in the next webinar that we do on the registration. And so the point is that what we were showing today is we take, the sections, they're all cut at the same, presumably the same angle. So there's an offset relative to the Allen Atlas space. And we could take a single, it's possible in the next, with the registration functions in NeuroInfo to take a single section and then to register it to adjust those angles that, the right, left, and dorsal ventral, all of and the rotations, all of those things into the Allen space. And then we can do that with a single section, or having put together this 3D volume from all of the sections put back together. Basically applies the same principles to that and registers all of them together. That will be covered in the next webinar.
That's great. One question, I got a couple histology type questions, and I've brought a couple of them, but one is, are there any citations you'd recommend for histology prep? And Chip, I think you have a current protocols that we could send a link around to, and that even includes, I think, a 3D printing files if they wanted to replicate exactly what you're doing with some of your holders.
Right.
Yeah. Okay.
And,
so I'll make sure that
can
in the
reach out to
email
me.
we send out, we send that as a reference. And I think another question is, we'll send out our original, one of the original papers, Brian, that we authored, that was in JCN, on how we developed this method and validated it. And that goes into a question we have about the accuracy and how much significant manual verification is necessary. And there's Chip's holder for specimens.
Yeah, so what we have the specimens where we can do 2 brains with this, and it's individual wells. We put the brains in, and then it fits into a dish so that there's minimal amount of volume. Anyway, so this is what we use.
And the 3D printing files are available online for those
3D
too.
printing of that.
Yep. So I'll make sure that we send out a link to your current protocols paper, Chip, and then our JCN paper on the development of the method, which goes through some validation as well. We are almost at the hour, and I don't see more questions coming in. So I really want to thank everyone, Chip and Brian. It was really a great informative webinar. I can't wait for the next one. And it sounds like we're getting a lot of questions toward that, so it should be exciting. And just as a reminder, we, don't forget to fill out the survey if you can. We'd really appreciate that. And also, this list of upcoming webinars has a lot of very interesting neuroscience topics. So I think with that, we can wrap things up. And thanks everybody.